Hello again. My name is Evan Norris from the University of Colorado, and it is with great pleasure that I'm able to present to you the long-awaited results of the notorious MRA pig study. Here are our disclosures. Recent in vitro work by Wilson et al. Has described the behavior of R1 and R2 star in blood, namely that it is very different than in plasma, particularly at the high gadolinium concentrations we see with contrast-enhanced MRA. These relationships, if correct, can help us accurately model and optimize contrast-enhanced MRA. However, they have yet to be validated in an in vivo model until now. This is an in vitro example of the deviation in R1 from linearity at both 1.5 and 3 Tesla due to the remarkably large amounts of water exchange across the red blood cell membrane, also known as the shutter speed effect. R2 star effects are more pronounced in blood than in plasma, particularly at 3 Tesla. Note that in blood, unlike in plasma, the R2 star is very similar for all agents. Furthermore, these effects are very relevant, with T2 star expected to be less than or equal to 2 milliseconds during typical first pass. Based on all this, we expect that at low injection rates, roughly less than 1 milliliter per second, the relative signal intensity increases in a quasi-linear fashion. This is then followed by a plateau phase and then ultimately a seemingly paradoxical decline, challenging the commonly held construct that the faster the gadolinium injection, the greater the signal intensity. Our goal is to validate a model whereby contrast-enhanced MRA signal intensity can be accurately predicted based on in vivo gadolinium concentration. In doing so, we aim to comprehensively understand the effects of R1 and R2 star in accurately predicting signal intensity. The results of our findings aim to validate the use of slower or more dilute gadolinium injections while performing contrast-enhanced MRA. Regarding our methods, 8 juvenile German pigs, which closely approximated small human size, were investigated on a 1.5 Tesla magnet. Anesthesia was administered and an aortic catheter was placed in order to collect arterial blood samples during time-resolved contrast-enhanced MRA. We investigated 4 gadolinium agents: gadobenate, gadoteridol, gadobutrol, and gadophosphate. Relevant relationships between R1 and R2 star and blood concentration for all agents, as well as protein binding coefficients for gadobenate and gadophosphate, were determined from doped blood using look-locker and multi-echo FFP sequences per the technique of Wilson et al. Continuing on, 2 pigs each received a total of 3 injections of the same contrast agent in randomized order, either at 1, 2, or 3 milliliters per second. Contemporaneously, arterial blood was sampled every 2 seconds and sent for mass spectrometry to determine the precise gadolinium concentration. This was then used to predict MRI signal intensity and compared with the observed signal intensity for each sampled time point. Prediction of signal intensity came from the following equations below, which were derived by Wilson et al. These take into account the shutter speed effect due to water flux across the red blood cell, protein binding for gadobenate and gadophosphate, and the R2 star lengthening effects in blood. Moving on to results. Just like in human blood, the nonlinear behavior of R1 in vivo is demonstrated in pigs. Similarly, R2 star in pig blood was much greater than in plasma and similar for all agents. This table shows that the protein binding coefficients, little r2 star values, and the unbound little r1 free values were essentially identical between humans and pigs. Little r1 bound values for gadobenate and gadophosphate, however, were much less than in humans, perhaps related to the species' difference in albumin. These doped pig samples for each of the contrast agents demonstrate how well the mass spectrometry performed, with R-squared values all greater than 0.99. These graphs demonstrate the results of 3 consecutive injections of non-protein binding agents. Gadoteridol is demonstrated on the top row, and gadobutrol in the bottom. The same relative scaling is used across all 3 consecutive injections. The red line is actual signal intensity by time-resolved contrast-enhanced MRA, and the blue line is the prediction. Note the excellent correlation including all baselines and tails across all 3 injections for both contrast agents. Here we show similar results for the protein-binding agents gadobenate and gadophosphate. This slide highlights what happens if you don't model the full R1 and R2 star effects. On the left is a repeat of the full predictive model for injections of gadoteridol and gadophosphate in full consideration of R1 and R2 star effects. You can see how the predictive model breaks down in the middle and right graphs if we neglect either R1 effects, demonstrated in the middle graphs, or R1 as well as R2 star effects, as demonstrated on the right graphs. This graph shows an average of the signal intensity versus injection rate at 1 to 3 milliliters per second for the half-molar agents gadoteridol and gadobenate. Note the red line shows very minimal increase in signal intensity as we increase injection rate. This is as predicted. The blue line shows the corresponding decrease in duration of the bolus, and shorter boluses act to increase blurring artifacts. In conclusion, the theorized sublinear behavior of R1 and the high R2 star values achieved in blood during first-pass contrast-enhanced MRA, as described by Wilson et al., are validated in vivo in an animal model. This should enable more accurate modeling that can optimize the use of gadolinium-based contrast agents for contrast-enhanced MRA. Thank you very much for your time, and I hope you enjoy the rest of the conference.